Friends, today we will see about DNA repair mechanisms. Previously, we have seen the DNA replication. So, as a continuation, in this video, we will see the DNA repair mechanism. So, what is the DNA repairing? Whenever there is a problem in the nucleotide arrangements, it will lead to some damage. So, those damages will be handled by the cells by themselves. So for that it needs some mechanisms. First, the cells need an undamaged strand of DNA template. Based on that, the damaged DNA can be corrected. Otherwise, if both these strands are repaired, for example in oxidative stress, both these strands will be damaged. So in those conditions it will use the sister chromatids to correct it. So the third is very important, which is the recovery mechanisms. So if they cannot correct by themselves, our cells have some repair mechanisms. So those mechanisms will come into the action and they will take care of those damaged cells. If there is a defect in that repair mechanism, then it is called as cancer. If a cell has some gene and some nucleotides are mismatched, those mismatched has to be corrected by themselves. If that repair mechanism is not working properly, it cannot be corrected, then it is leading to cancer. So what are those DNA repair mechanisms? So the types: first it is the mismatch repair, second it is base excision repair, and third it is double stranded DNA repair, and fourth it is repair of damage caused by UV light. So in this video we will see only about mismatch repair. To make the video as a short, I am going to make it as a fourth thing. So today we will see only about the methyl directed mismatch repair. So here you can see a daughter strand and one parent strand. So this is the daughter strand. Always the daughter strand or a newly synthesized strand will run in 5' to 3' direction. So this is the parent strand, which is 3' to 5' direction. So as an example, I have mentioned G, that is guanine, in daughter strand and thymine in the parent strand. So is G and T are hydrogen bonded? No. Always the thymine will pass with adenine and guanine will pass with only cytosine. The bond between the adenine and thymine is double hydrogen bond. The bond between cytosine and guanine is triple hydrogen bond. All this we have already seen in our previous video. So now I have mentioned one more thing. It is GATC sequence, which is methylated. And here also you can see a GATC sequence with methylated. What is it? How it is important? So the GATC sequence will occur every 1000 nucleotides. So each and every 1000 nucleotides we will have GATC sequence, which is methylated at its adenine position. So this is very important. The bond which I have mentioned is at adenine. So it is adenine methylated GATC sequence. So how it is important in mismatch repair? So for repair mechanism, it is a protein. All repair mechanisms are proteins. So here it is mute protein. So this function of the mute protein is it will recognize this methylated GATC sequence. So what this GATC sequence is telling? So this GATC, which is methylated, it means it is the parent strand. So if it is there for a long time, then only it can get methylated, whereas the daughter strands and all hemimethylated or non-methylated strands. So it will recognize the parent strand and it will tell that the function is going to occur in the daughter strand. So now the DNA endonuclease and DNA exonuclease will come into the action. The DNA endonuclease and exonuclease will nix or cuts the strand and removes the mismatch or the mutated nucleotide. So now here it is empty. So what is the next action? The problematic has been removed. So now we have to repair with the correct sequence. So DNA polymerase function is to chain elongation. So the function of DNA polymerase is for elongation, and we have the parent strand here with the template. So based on this, DNA polymerase is going to elongate the chain with the correct nucleotide bases. So once it has been formed, these ends, the old strand and the newly synthesized short chain has to be ligated, which is done by DNA ligase. Hence at the end we will get a new daughter strand which is corrected based upon the template of parent strand. Hence what I have to tell at the end is, or if the summary if you are asking, it is the function of DNA endonuclease, exonuclease, DNA polymerase and DNA ligase with help of mute protein will recognize this damaged or mismatched nucleotide, and it is removing off and forming a new correct sequences. So this is methylated directed mismatch. This kind of repair mechanism, if it gets mutated or if this repair mechanism is not working properly, it will be seen commonly in colon cancer, which is called as hereditary non-polyposis colon cancer. But in that cancer population, only 5% will be due to this mismatch repair mechanisms. Other will be due to some other causes. Okay, in next video we will see the other repair mechanisms. Thank you.